Ultrastructure of the mitochondrion. This is the structure of the mitochondrion, which you will see using a transmission electron microscope. Since for it can see structures that cannot be observed using a compound light microscope. Now what happens is that this mitochondrion here is the site for aerobic respiration. Respiration can occur in a cell without a mitochondrion. For example, red blood cells lack a mitochondria. Occur if it is to occur there, then it should be anaerobic respiration. When it comes to aerobic respiration, it occurs, most of it, part of it occurs in the mitochondrion. So with aerobic respiration, mitochondrion is a must. However, its ultrastructure is recommended to be understood by candidates of new curriculum A level. This one here, you see here, is the outer membrane. This one here is the outer membrane. That outer membrane is highly porous. It allows many small molecules to easily go through. Okay, they easily go through, especially molecules that are below 5,000 Daltons. Below 5,000 Daltons, they can easily go through this mitochondrion. A Dalton is a hydrogen atom. So molecules below 5,000 Daltons can easily go through the outer membrane of the mitochondrion. Now, so it is not selective. It is highly permeable. Almost everything can go through so long as it is that small. Now what happens is this outer membrane is about 6 to 7 nanometers thick. Of course, that is very important when it comes to diffusion, because materials normally, some of materials go through by diffusion, and remember diffusion is enhanced by a shorter distance. However, even transmembrane proteins that facilitate diffusion will also permit easy movement of materials across it. So that small size is very, very important, about 6 to 7 nanometers thick. So what happens is that here we have the intermembrane space. This intermembrane space is about 10 to 20 nanometers wide, that intermembrane space. Now this membrane here, this intermembrane space here is very important because it contains a lot of proteins. For example, enzymes involved in aerobic respiration. There is a lot of enzymes. Now, it sounds as if I'm discussing structure and function, which is still okay. However, don't forget that these enzymes are also part of structure. They are also part of structure. They occupy space, so they are also part of structure. So these enzymes, these proteins are located there that facilitate aerobic respiration. But also, it contains a lot of protons. So a lot of proteins are highly packed there. Okay? A lot of protons are highly packed there. Don't forget that these protons are very important when it comes to ATP synthesis by oxidative phosphorylation. Now, we have the inner membrane. Now, this inner membrane here, this membrane is selective in nature. It is selective in nature. It does not allow many materials to go through. Some materials will never go through it. Others can easily go through it, so long as they pass through particularly designated channels, especially those ones which are hydrophobic, which are hydrophilic in nature. Yeah. Now, this membrane here, the inner membrane, is about 75% proteins. It is having a lot of proteins. We know membranes, plasma membranes, as phospholipid bilayers. Now, if you talk of a membrane having 75% proteins, meaning lipids and other whatever taking only 25, it is somehow not in line with the theoretical biology we know. However, this is what you have to know. This membrane here contains what you call the electron transport chain, ETC. This electron transport chain, most of them are proteins because they are enzymes. Okay? It is made up of other materials which are not proteins, but most of them are proteins, as we shall see in the future. So that one is also part whatever. So this membrane here is selective in nature. Selective in nature. Remember the outer membrane we said it is highly permeable because of the presence. It contains a protein called porine. Porine that forms pores in it, which allows easy passage of materials through it. Okay. Now, this is what we call crystal. This is what we call crystal. Crystal. Crystal for singular, many crystal. But this crystal, these are crystal you see here. However, one of them, I have identified one of them, and really there are some other structures. These structures you see here are the ones we call stocked particles. Stocked particles. Stocked particles, these stocked particles, they are part of the enzyme ATP synthase. It's a protein that conducts oxidative phosphorylation to form ATP. Okay? ATP synthase, having two domains. It is having the F1 domain and the F0 domain. The F0 domain is embedded into the inner mitochondrial membrane. The F1 is outside, having a bulbous head, which facilitates formation of ADP by moving as the protons are going through. Then the kinetic energy is coupled to combination of ADP and inorganic phosphate from ATP. So this is the stocked particle, but the whole of it, F1 and F2, the stocked particle is the enzyme ATP synthase. Now, the electron transport chain, I've already said that it is part of the inner mitochondrial membrane. All those ones are proteins. So don't get shocked to find out that 75% of the inner membrane are proteins. Okay. Now, this structure you see here, this structure you see here is the mitochondrial DNA. Mitochondrial DNA. However, this mitochondrial DNA is similar to ours. Is similar to our cells' DNA. Remember, mitochondria is within our cells as a host almost. There's what called endosymbiosis. Now, it has its own DNA, mitochondrial DNA. Now, this mitochondrial DNA, you see here, it is similar to our DNA, to the DNA of our cells, our eukaryotic cells, in that it is also having two strands that are antiparallel and forming a helix. So it is double-stranded. So it is governed by the same genetic code that governs our DNA. Though there are some technical slight differences with it. For example, when it comes to non-sense codons or stop codons, there is some slight difference there, a little bit. Now, what you see here, that one is a ribosome. It is a mitochondrial ribosome. There are many mitochondrial ribosomes. There are many. This ribosome here is made up of a small subunit, which is 30S, and a large subunit, which is 50S. 50S, S is standing for Svedberg's constant. These are sedimentation whatever units of sedimentation based on density. So 30S and 50S, meaning that if we add 50S plus 30S, we shall end up getting 70S. Forget your 80S. So now that one is the ribosome there. Now you all know that ribosomes, what ribosomes are involved in. They're involved in protein synthesis. Now, what you see here, it is the matrix. The matrix. Now, this matrix here is semi-fluid. It is semi-fluid. Semi-fluid. By semi-fluid, its semi-fluid nature creates a very good environment for possession of very many inclusions there, including coenzymes such as NAD. NAD is there, both oxidized and reduced NAD is there. They are there, okay? They are there. There are enzymes involved. There are enzymes involved in very many biosynthetic reactions, occurring there. So therefore, this one, there is a lot of whatever there in that matrix there, mineral salts, most of them acting as enzyme activators, coenzymes such as NAD. They are there, very many of them, they are there. There are lipids there. You will find lipids there, inside there. There will be lipids, they will be found there. Also, you will be finding there carboxylic acids. Carboxylic acids you will find them there. Carboxylic acids will be found there. You will find there ATP. You find there ATP. You will find there ATP, of course, because it is a product made. You will find there ADP. Then you find there also inorganic phosphates. They are concentrated there. So that one, what we have been discussing, is what we call the ultrastructure of the mitochondrion. Most of those inclusions cannot be seen using a light microscope. For example, the ribosomes, the DNA, the stalked particles, most of them came to be understood by scientists after following the invention of the electron microscope. That's when these whatever were seen, probably about around 1935. It was invented already, and these ones came to our knowledge as scientists. So that is the ultrastructure of the mitochondrion. Feel free to comment, to ask questions, to bring contributions, to raise concerns where you have found concerns, and also follow my channel, BiologyKingUg, on YouTube, and also BiologyKingUg on TikTok. But on TikTok, I put there have videos, full videos. You will find them on the YouTube channel, BiologyKingUg. Call your friend, your brother, your sister, whoever who does biology, or anyone who has passion for biology, to follow my channel. More things are coming. I'm also increasing technology. Vision will be okay with time, and we shall be enjoying videos. Thank you very much.